Today we are demonstrating a simplified technique to excise corneal and to eviscerate retinal tissue from a human ocular globe of cadaveric donors. The technique described here will help to excise good quality tissue to be used for transplantation, surgical or research purposes without damaging other tissue of the ocular globe. With 18 years of expertise and hundreds of corneas transplanted each year, we have maintained a good standard of our work by developing new techniques and improving our skill every day. To achieve this, we excise the cornea first, followed by retinal evisceration.
The cornea is an avascular tissue which enables the transmission of light onto the retina, and for this purpose should always maintain a good degree of transparency. The cornea comprises 5 layers: the epithelium, Bowman's layer, stroma, decimus membrane, and endothelium. If the cornea is damaged, it can be replaced with the help of a suitable donor cornea. The choroid is the intermediate tunic between the sclera and retina, bounded on the interior by the Bruch's membrane, and is responsible for blood flow in the eye. Appropriate evisceration of the choroid is as important as removal of retina. The retina helps to receive the light from the cornea and lens and converts it into the chemical energy, eventually transmitted to the brain with help of the optic nerve. As it is transparent and delicate,